in the water, the synergetic influence of all compounds. So when I modeled the ratio, I had a good model. So it is in the what you get in the end. This is what you want to model based on that.
Yeah, but I would also expect that there are synergistic effects happening, especially since here in Switzerland we have dozens of chemicals in the water, including glyphosate. So maybe it would be very interesting to create also the analysis with glyphosate.
Yes, Felix.
Would love to
Just
see this Aquaphotomics glyphosate toxicology study being done.
Yes, just do it. I mean, glyphosate is a big problem. Absolutely.
I hear about that compound for the first time, like Petia. Now, first time. So can you even please, Felix, write the name of the? I can't. I don't know how
it's. Your email, yeah.
Thank you. Thank you.
You write Stephanie Senef, and she will tell you everything about glyphosate.
Oh.
Yes.
I didn't know. Any questions about this topic? More? Then I would like, we don't have much time, but I enjoy discussion, and I wanted to ask if person David Tjandra Nugraha is here, because we just mentioned something with regard to stress, and he has a poster which is actually connected to that topic.
Hello, I'm here.
Petya, thank you so much. This was wonderful.
Yes, good day, colleagues, and thank you for the opportunity. And thank you very much for the discussion. And as a master student, this is a very fortunate opportunity for me to learn so much from the experts of this world. Thank you.
So, David, you attempted at a quantification of ascorbic acid.
Yes.
Yes. So, why were you interested in this compound? Can you tell us what does it say about the mung bean sprout, the quality of this?
So actually, the idea of this research was we were worried that there was no actual standard for mung bean sprouting. In industries, they grow it in various variety of time, for example, 3 days, 5 days, and there was not really a certain set amount of time. So that's why we try to follow the germination process, the biological process through the NIRS and Aquaphotomics, and in a set amount of interval, especially 6 hours of intervals, and we saw some interesting pattern within the pin changes, and we try to make a regression model to predict the germination time, and also as an qualitative indicator, we also try to quantify the ascorbic acid content, and as it is quite in a small amount and within the ppm amount, I think it's around 10 to 50 ppm of the ascorbic acid. And within this kind of a complex matrix, we would like to see if the NIRS and Aquaphotomics could also quantify this organic compound within such low concentration. And we compare it with the conventional technique, and we succeed to create a model which has which agreed to the results from the conventional technique.
Can I ask something?
Yes, yes. Can you please also show results, so David?
Yes, yes.
Thank you.
Do you remember what is the spectral pattern of germination time? Of the expression vector of the germination time? Which are the bands that are related to the germination time? No, you look at your aquagram, because I cannot read very small letters, so I cannot. So that's why I'm asking, which is the spectral pattern that is related to the germination time?
From our investigation, we suspected that within the early 1500s, so the strong water pond and the NH pond, and we suspect that there might be changes, especially during the enzymatic processes from the sprouting, and also there are less forming of the weak water pond structure due to the biological process. So this is a preliminary result, preliminary screening, and it just we just try to see if it has the potential to follow the process.
Could you please tell me about the legend so I can read them? What is the black and red and yellow in your A aquagram?
In the A aquagram, the black is the ascorbic acid content. Okay. It is the lowest concentration, the 0 ppm, and the red is 10 ppm, and the yellow is 20, 30. Okay. Yes.
Then you increase the concentrations, all right. And the aquagram which is below?
It starts from the 0 hour of the germination time. So
okay,
0 hour, and the smallest, the largest, the blue lines is the one.
Yes.
Well, the longest sprouting time.
Yes. Okay. Thank you. Perfect. The last aquagram, the last spectral pattern, is a typical spectral pattern of a cell, of a viable cell. So that's why I asked the question. So you start measuring. Once you see this aquagram, you know what you're measuring. It's beautiful. It's really beautiful.
Thank you.
And vitamin C in the aquagrams that I can see, very much concentration dependent, the spectral pattern. So we people, we think that when we take a lot of something, it will be very good. No, it's not the case. So sometimes very small quantities that do the work. So, and vitamin C is a beautiful example of that. So you can, with different and not only vitamin C, but especially different concentration of vitamin C, you can play and you can get different aquagrams, which means different functionality of the water. It's amazing substance. With the model, I liked with the model for vitamin C, when you could measure the vitamin C in a system, in a different system, right? So have you tried different systems, not the sprout only, but other systems with that particular, the same model?
Yeah, so that is an interesting point of view that we haven't tried it actually in a different system. So we only tried it with the calibrated pure ascorbic acid component and immediately in the mung bean sprout. Yeah. So that might be some way to verify whether this model is potent enough to quantify that organic acid.
Yeah. But it's very good work. Thank you.
Thank you for the interpretation.
Thank you, David. I just wanted to add something because these are phenolic compounds. People nowadays in biotechnology make large investments into the artificial developments of these kind of compounds, which are then sold as supplements. But the point is that phenolic compounds are actually produced in response to the changes in the organism. So, for example, people stress bacteria or yeast in order to induce that kind of response, and then, to damage them basically, and then they produce different phenolic compounds. So what I saw, we had some study about that, and what I see from your aquagram, what we have when we tried to measure the concentration of this phenolic compound in our study, we had 1460 nanometers band, totally related, and it followed the increase of phenolic component. So I think this is what your Mung bean is showing you also. So we have an agreement. We also had a nice correlation with 1426, but it may be related because we worked with yeast, so it's a bit different organism. So I think might be. This is beautiful. I really like this. Thank you for being here and answering my question.
Thank you also for the suggestions. It is very valuable. Any
questions maybe for David? So if there are no questions for David, I would like to relate, because it is connected to this topic. Is Anastasia here?
Hello, everyone.
Hi, Anastasia.
I'm really here